Autoimmune conditions are complex disease states where inflammatory and immune dysregulation overwhelm numerous regulatory systems. Their development and severity are often influenced by a legion of factors including genetics, immune dysregulation and infections, intestinal hyperpermeability, dysbiosis and toxicity, nutritional deficiencies, and stress. Bioceuticals is proud to present the Reprogramming Autoimmunity Seminar Series in November 2016. The aim of this seminar is to delve deep into the known imbalances seen in autoimmune diseases and to learn the modern integrative treatments which can improve the health of patients suffering autoimmune-related illness. You will leave the seminar confident in assessing the complex imbalances seen in various autoimmune disorders, prescribing safe herbal and nutritional medicines to combat immune imbalances, and recommending effective nutritional and lifestyle interventions for the management of autoimmune disease symptoms. Your presenter, Belinda Reynolds, is a dietitian and senior educator for bioceuticals who has shared her wealth of knowledge across Australia and New Zealand. Join Belinda at this half-day seminar throughout November 2016 to learn more about the evidence-based approaches for rebalancing immune dysregulation and how to enable your patients to enjoy a more fruitful life. Register now for this important education event at bioceuticals.com.au/education/events.
This is FX Medicine and I'm Andrew Whitfield Cook. Joining me on the line today from Melbourne, Australia is Dr. Denise Furness, who conducted her PhD in Nutritional Genomics and Genome Health at the Commonwealth Scientific and Industrial Research Organisation, that's the CSIRO. Everybody in Australia just calls it the CSIRO, a Department of Human Nutrition under the mentorship of Professor Michael Fenwick. She's been involved in genetics and nutrition health research for over 12 years and was principal investigator in her postdoc study Predicting Adverse Pregnancy Study, which is the PAPO study, at the Women and Children's Hospital in Adelaide. Her interests include the diagnosis and treatment of underlying triggers such as detoxification, inflammation, oxidative stress and methylation. Denise works closely with integrative practitioners in order to apply this knowledge and address the possible underlying issues associated with various health concerns including fertility, gastrointestinal disorders and mental health. And I warmly welcome you to FX Medicine. How are you, Denise?
I am well, thank you, Andrew.
And I should have added into your bio, you are the loving wife of your husband and the loving mother of one child and one on the way.
Yes, we have a little boy who's just turned 4 and one that's probably about 2 months away. So yeah, counting down the days.
Yeah, counting down the days. Now, you've got a heck of a lot of expertise in genetic sampling. Can you do me a favour? Can you do us a favour? Because you've done a lot in your very young life. Can you take our listeners through where you started? What was your initial interest in nutrition? And then take us through your training and genetics, training into genetics.
So it started for me really in science. So I did my first degree, undergraduate degree, was in science, biological science. And then from there I went on to do an honours project, and that was in conjunction with Deakin University, where I did my undergrad degree, and CSIRO, the Australian Animal Health Laboratories. And that particular project for my honours was really around molecular biology and genetics. And then that led into a job with the CSIRO. And that was really a bit more sort of hardcore genetics. And I loved it at the time, but there was a lot of animal work. And to be honest, I didn't enjoy that. Even though I loved the experience, anyone that knows the Australian Animal Health Laboratory, CSIRO, it's a pretty amazing place to work. It's a fantastic facility, but and I love the genetics, but the animal work didn't get me. So I started looking around for a PhD, and the advice I'd been given was make sure you love it, because reality is you're going to be doing this for a while and you need to have that sort of passion and drive to complete a PhD. So I was thinking, well, how can I utilise my interest in genetics, my experience in genetics in an area that I think I want to be in sort of forever. And I learnt about this particular field, nutritional genomics or nutrigenomics. And I was quite excited about this sort of area of understanding genetic testing that can influence, your diet, or how certain, nutrition can impact on how well your gene functions and sort of health. So in myself, personally, I'd always, I guess, been quite passionate about health. I was into nutrition. I've always been an exerciser. So I sort of had that personal area of interest.
Yeah.
So I thought this would be a great way to combine my experience in genetics with sort of, I guess, my own personal interest. So I applied for a PhD at CSIRO Human Nutrition, and this was in Adelaide, under Professor Michael Fennick, who's known to be, I guess, the guru here in Australia with nutrigenomics.
Yeah.
Absolutely. So I was very lucky to get a government scholarship, a CRC, federal government scholarship, and I moved to Adelaide to do a PhD in his laboratory, and I was there for 4 years. And this particular project was in relation to nutritional genomics, so that interaction between the genes and the diet, particularly in relation to pregnancy outcomes. And then from there, I was awarded a postdoctoral position with the University of Adelaide and the Women's and Children's Hospital in Adelaide. And then I went on to conduct the study that you mentioned at the beginning in the bio, and really continued that research from CSIRO, but broadened it, because initially it was really more about folate nutrigenomics, and for those that have heard about MTHFR and B vitamins and methylation and sort of DNA damage, whereas when I went into my postdoctoral position, we then really expanded it to more than just that sort of B vitamin metabolism, that gene interaction around that pathway. We moved into vitamin D and inflammatory markers and oxidative stress and things like that. So that's really my training and expertise. In that time, of course, anyone that's into this area would know that you're always studying. So I have done some, when I was on maternity leave last time, I thought, I'll just do a degree in, human nutrition and exercise physiology, which I got to third year. I haven't totally completed, but I also have a Cert 3 and 4 in fitness, and I've worked in the fitness industry, and I've done multiple certificates in nutritional medicine and other courses like that. So there's been a lot of training, I guess, and working in the area for the last 15, possibly longer, years.
The
last 4 years, I guess, I'm working in the clinic. So that has moved from research now to clinical practice.
Right. I think when we're talking about these fields of genetics, the genetic, genomic and nutrigenomic testing, and that's what we're going to be discussing today on FX Medicine, I need you to clear some things up for me, because I got to say I feel overwhelmed. So I get very confused as to what is their usefulness, what are they actually doing when they're testing each of these things. Can you clear up or differentiate for us, and indeed me, what is the difference between genetic testing, genomic testing, and nutrigenomic testing?
All right. So, I was going to say it's a fairly easy one, but hopefully I can communicate
for you,
well.
Maybe.
So genetic testing, genetic testing, really you're just testing the gene, something within that gene, perhaps a variation in the gene or the copy numbers of that gene. So it's literally just about something to do with the gene. When we say genomic testing, we're thinking about the genome. So not just one gene, we're thinking about all of the genes that would make up, say if it's a human genome, or if it was a different animal or plant, the plant genome. So the gene is, we're just doing some testing in relation to one gene. The genome is that sort of complete set of genes. So you'll be doing some type of testing that can impact on the genome, or you're doing, like, for example, if you talk about the Human Genome Project, they were trying to determine all of the genes. And then when we talk about nutrigenomic testing, this is really an area that is looking at genes that are linked with our nutrition and our lifestyle. So nutrigenomics is really discussing the science of the interaction between our genes and, in particular, diet, but also incorporating things like toxins and chemicals and things like that. So you're still just testing the genes, but you want to know the purpose of testing them, or the reason you're doing it is because you want to know more about this interaction between the genes and the environment. And that's really based on research that's been done to tell us, for example, we know that MTHFR impacts on folate metabolism.
Yep.
So that's sort of nutrigenomic testing, looking at that interaction between our genes and our nutrition or our lifestyle. And I will just make one comment. We say nutrigenomic testing,
Yep.
and that's probably the most common way of describing it. But often when we're talking about nutrigenomics, if you wanted to get really technical, we're often really talking about nutrigenetic testing,
Right.
because we're testing a gene,
Yes.
not the whole genome.
Right.
So, but the common term is nutrigenomics, and the field is called nutrigenomics. So that is why you'll hear that more often.
It's kind of like how people are talking about the microbiome when they really are referring to the microbiota.
Absolutely. Yes.
Yeah.
Yes.
But it still
Yes.
confuses me, because now we're going to be talking about SNPs. So what exactly are we talking about when we're talking testing of SNPs? And is this genetic testing like that which is done for, say, the original disease, the poster child, dare I call it that, the tragic disease called Huntington's chorea or Huntington's disease? Is that the same as testing for SNPs?
First of all, I'll describe what SNP testing is. So SNP testing, SNP stands for single nucleotide polymorphism. It's a big word. The listeners don't really need to know what it stands for. All it means is that we're testing a gene, so it's a type of genetic testing, and we're looking for a single variation. So our genes are made up of our DNA, and our DNA is made of 4 bases, or just 4 letters, and it's these letters in combination that make up our genes and our whole genome. So when we say a SNP, we're just talking about one change within one of those letters. It's a very, small change within the gene. So that is SNP testing. When we talk about Huntington disease, so basically that is genetic testing.
Yeah.
So first of all, I'll say I don't test for diseases. The type of genetic testing I do is I'm often looking at the SNPs, what you were talking about, changes that might affect the function of a gene or how it interacts with a nutrient. So Huntington's disease basically is a gene test, but you're looking at, to my understanding, if I recall correctly, it's actually to do with the number of repeats within that gene. So you're not looking at a SNP, you're looking at repeats, because there are different within the gene. So it's still a genetic test, but we're not looking for one single base pair or one letter difference. We're looking for a number of repeats.
Yeah.
So there are different types of genetic testing, in that some could be SNP testing, what I just described. We can also look at things like insertions or deletions, or the number of repeats, or even copy numbers, so the amount of genes. So there are different forms of genetic testing.
So I see now that orthodox medicine is getting very interested in SNP testing when they're looking at how somebody will respond to a certain drug therapy. And, I think the first one I ever heard about was how a patient might respond or not respond to the drug tamoxifen after having breast cancer. There are others, there are warfarin and things like that, but it's mainly the pharmacotherapy that they're trying to modulate to see if somebody will respond to a drug therapy. And therefore, they call that personalised medicine. But that differs wildly from what we're talking about today, and that is nutrigenomic testing. So how a SNP and a nutrient or a diet interact. So I got to ask, is this a case of turf war, or are they wildly different? Are they really different?
So the area, when we're talking about the genes and sort of genetic testing that can influence possibly the benefits or side effects with drugs, that area is actually called pharmacogenomics, as I think you just mentioned.
Yeah.
So pharmacogenomics and nutrigenomics are different in the sense that pharmacogenomics is obviously relating to the gene-drug interaction, whereas our nutrigenomics is talking about that more sort of nutrition lifestyle interaction with the genes. So I don't think it's much of a turf war. I would like to think that myself as a scientist or allied health practitioners really work with the doctors. So the way that I practice, actually, I work in a medical practice with 4 GPs, and we really all work together, and they often get me to sort of interpret the genetics, and we really often, refer back and forward to sort of get those optimal cases or optimal outcomes for the patient. However, in the sense of the pharmacogenomics really being related to drugs, it's more likely that this information is going to be delivered or should be delivered from a medical practitioner or a specialist, such as a psychiatrist, because we know a lot of these genes also impact on the success of a lot of the mental health medications as well. So in that case, I think we will see that this kind of testing, pharmacogenomics, is more going to come from the medical doctors and the specialists, because they're the ones that are advising the drugs. And in the case of myself being a scientist and also a registered nutritionist, I'm not going to prescribe certain drugs. However, I can still talk about those genes with the patient, but you definitely want to be working with the doctor.
Right. So to me, that does seem like a case of, dare I say, I mean, that's a bit of a flippant term to say turf war. It's more of a paradigm of practice. Doctors prescribe drugs, and so therefore pharmacogenomics, which should, when you say the word, should really be pharmacogenetics.
Yes,
versus,
you're learning. That's great.
Versus somebody who is more into, we say the word as a broad brush, saying natural medicine, talking about nutrigenomics, because they're dealing more with health and diet rather than disease states.
Yeah, and I think generally that is right. Obviously, the doctors I work with are integrative GPs, so they're a little different. They have a strong interest in nutritional medicine and lifestyle medicine, so they also do a lot of nutritional genomic testing. But I would say majority of doctors that don't have the time to sort of, or the passion to sort of get into the nutrition, they're really the ones that are going to be doing the pharmacogenomic testing, and it's the other practitioners that are mainly focused in nutrition and lifestyle that would do that nutrigenomic testing.
Yeah. Can you take our listeners through what it means to have a polymorphism of a particular snip? Polymorphism. There seems to be this whole school of thought that, what it means when you go on to develop certain disease states, as opposed to professional advice you can give to listeners on interpretation and understanding of certain results when you are shown to have a particular snip. What does it mean if you have a certain snip?
So just reiterating what I mentioned before, a snip is a single base pair change. So it's a change within the gene, and we often call this a polymorphism. So poly meaning many and morph is sort of different. So just sort of showing that there can be a difference in this base pair.
Yeah.
So what happens when you identify a SNP, and I will just mention that not all SNPs or genetic variations cause a functional effect or associated with disease. Some SNPs actually, you will still get a normal protein or an enzyme being formed. But we know now that many SNPs, and this is based on medical research, clinical trials, genome-wide studies, we know that many SNPs actually can impact on how well the protein or the enzyme or the receptor, whatever's being coded by that gene, how well that functions. And then this may impact on the person's health. What we cannot say is that if someone has, and I'll use MTHFR again simply because it's a common one and we've been doing research on it for 20 years, a lot of people sort of know about it. But if someone does have a genetic variation in MTHFR, this does not mean that you are going to, or this is the cause of your fertility issues, or that you're going to have a mental health problem, or you're going to get cardiovascular disease. All it means is that you don't metabolize folate as well, and this could be one of those underlying factors linked with whatever the person's issue may be, along with we'll be looking at a number of other markers as well. So a number of genes, looking at the nutrient levels, looking at a number of things. So the genetics is an important tool. I believe it's an important tool as part of that total holistic approach. So you can't say that if someone has a SNP, they're going to develop a disease. But what we can do is educate them about how well their body is working, or how well their body processes something like folate that has been linked with, say, pregnancy disorders. And then from that information, we can educate the patient again on what might be the best diet. Okay, you might need more folate. Let's talk about your leafy greens. How many serves of vegetables are you getting a day? If they've got certain other genes, maybe they're not metabolizing a certain folate well, let's talk about introducing a different form of folate. So it's not necessarily directly to disease, but more about things that can relate to adverse health outcomes.
Yeah. I think when you're talking about, we always use that poster child of MTHFR. So that's the methylene tetrahydrofolate reductase.
Well done.
Yeah, I have a memory. When we're talking about that, let's take that as a snapshot and let's go back in time to before folate supplementation, mandatory fortification in foods, in breads in Australia. So if we went back to that time where if we look at the prevalence of the various SNPs, and there are a range, we'll talk about that later. Indeed, we might talk about that in a separate podcast. But when we're talking about these SNPs regarding or referring to folate metabolism, if you wanted to take that snapshot of, you've got a certain prevalence of these SNPs in the population, certain ones will work well, certain ones mediocre, certain ones poorly. If you then look at health outcomes of that group, regardless of any supplementation, this is before food fortification, then you can then say, even if you had that gene SNP, most people, many pregnant ladies, didn't have an issue with their baby. So therefore, they were having enough of the natural folate to be able to allow that gene to function properly. But those people who had the SNP, they are at increased risk. And perhaps these are the ones that ended up with a baby with a neural tube crest defect or a cleft palate. Is that the right thinking?
Yes. So you are right. There are many people in the population that would, because, for example, the MTHFR, 677 that we're talking about and 1298, these are very common polymorphisms or genetic variations. And there are many people that have that had babies before the mandatory fortification of folate and had healthy outcomes. However, it is important to note that it's likely that they were taking a prenatal that had folate and other B vitamins, because we do know that those with the MTHFR gene are at much higher risk for neural tube defects and congenital abnormalities. So that's been shown in a number of populations. But you are completely right when you were talking about the diet. In those that actually are eating a real food diet, because folate's found in lots of foods, green leafy vegetables, legumes, particularly lentils, fruits, nuts. So if someone's eating a good diet full of real food, it's likely they're getting enough folate. The issue is that in our day and age, most of us are not eating enough of the vegetables and the real food because we are busy, we're on the run, we're time poor, everything's packaged and processed and quick and easy. And a lot of these sort of nutrients are much lower than what would have been in our real food. So those that are eating the healthy foods, as you said, like the green leafy vegetables, are likely that they didn't have the complications. So you're exactly right. That's really that whole area of nutritional genomics, talking about the interaction between the genes and the nutrition.
So when you referring back to your PAPO study, the Predicting Adverse Pregnancy study, did that study look at potentially those people that had poor diet? Were they at increased risk of, say, MTHFR issues like cleft palate?
Yeah, so our study, we were working with two populations. One was at the Women's and Children's Hospital in Adelaide, which is a relatively, I wouldn't say affluent suburb, but it's a good suburb. It's in North Adelaide. It's still a public hospital though, so you're getting a wide range of patients. But we also recruited another large study I was involved in. I wasn't the primary investigator, but was involved in something called the SCOPE study, the screening of pregnancy endpoints. This is a very, very large study conducted in Adelaide and New Zealand and around the world. We all got together, and I think we recruited about 1,600 women, and that was from a hospital called the Lyell McEwen, and that is in a lower socioeconomic area. And we saw a lot more complications, pregnancy complications, in this particular population. And that is because obviously diet was poorer, but there was even women are still smoking. There's a lot of younger women getting pregnant. And, we definitely saw a much higher frequency of particular pregnancy complications in that population. And we also saw more of those sort of interaction with the genes. Because one of the things with genetics, and sometimes it's not as clear as we think it is because we don't often see the same results repeated in multiple studies. So we might do a study and say, we've definitely seen a strong association here. But because these genes are involved in that interaction between the environment, you then go and repeat the study in a different population who are living a different lifestyle, or particularly a different country. So some of our work we repeated in New Zealand, that we were fairly certain of particular genes, but we didn't see the same things in the New Zealand population, and that's because when we looked at the ethnicity, there was the particular hospital, there was a lot of Maoris too, so it's a different ethnicity, so we're not seeing the same frequency of the genes, plus their lifestyle is completely different.
And this is one of the issues. I mean, you could transpose that exact same argument onto work with probiotics. If you're feeding these bugs with a certain food, and just likewise, if you're feeding genes with certain foods, then if you're talking about a certain gene snip having a prevalence for a certain disorder in, let's say, I've seen this pop up heaps of times, the Han Chinese population. Well, that may well be a Han Chinese population in China, not a Han Chinese population in Seattle. You know, you know what I mean?
Yes.
And where they've got a different dietary influence.
And just on my own personal experience sort of getting back to MTHFR too, even though I have no doubt in the significant effect of that genetic variation, because we also did, when I started at CSIRO, we did a lot of cell culture work. So where you were actually sort of looking at, these genes and you're adding amounts of folate and B12 and DNA damage and the effect of MTHFR and things. So we know that with that particular SNP or variation, the enzyme activity really is significantly reduced, that you're going to get more DNA damage, you're going to get all these kind of things, particularly when in the particular cell culture, when we're talking about the media, if it had low folate and sort of B vitamins and things in this sort of, I guess, scientific way that we were doing it, sort of mimicking what would be going on in the body. But then when we're talking about the PAPO study, the women's and children's, I actually didn't see an association. To be honest, I was almost a bit disappointed because of what's known with MTHFR, but in my pregnant population. But then when we discussed it with my colleagues, it actually made perfect sense because our women were all on prenatal supplements,
right? And
some of them, because of our school of thought, and at the time, my supervisor, Professor Gus Decker, who was sort of the head of maternal fetal medicine at one of the hospitals, and the other wonderful obstetrician I worked with, Dr. Dee McCormick, we were very passionate about B vitamins. They knew about the genes. People that do research in this area have a good understanding. So, these doctors are informing these patients that, okay, we know you've got these SNPs. It's probably better that you be on, say, a higher dose of folate. So actually, our study was actually a little bit sort of biased in the sense that majority of our women were probably on even higher doses of B vitamins than what a standard, a woman outside of our study would have been on. So we didn't see the association with MTHFR, but our women were on high-dose B vitamins. So we probably hid that effect.
Yeah. Did you see a reverse association? Did you see?
We actually had good. So particularly in the recurrent miscarriage clinic, we had good outcomes.
Yeah.
So we had improved outcomes, which is what you would hope. But some of the things from a research perspective, you want to see certain associations, and we didn't see those because I guess we were sort of treating, and the research was slightly biased because we all believed in the power of bees for particularly those that had. And I'll also say it wasn't just MTHFR. We were looking at methionine synthase reductase, MTHFD1, a whole host of genes within that methylation pathway. And if patients had those, we would then sort of talk to them and say, okay, you're likely to need some more Bs. B6, B12, folate were the main ones we would talk about.
So it's an unintentional intervention trial.
Yes. We actually wanted to have a randomised control trial, but we couldn't get through ethics.
Gotcha.
Because, you're not going to tell any pregnant woman not to have folate.
That's right, yeah.
We were going to do folate versus a B complex. But actually one of the obstetricians that I mentioned, Dee said she was the main obstetrician in the recurrent miscarriage clinic. Even though we were going to try to sort of do randomised and blind, she said, I don't know if I want to be, when I believe that the complex is going to help, I'm not sure. So, it really is quite hard for a researcher as well, particularly when we do care about patient outcomes as well.
So the only way to really look at that would be to do a prospective trial, wouldn't it?
Yes.
And then you're outside the ethics of having to supplement.
Yes, exactly. And I guess what would be, even more interesting is looking at sort of in populations where perhaps people aren't supplementing with folic acid too, and then looking at the difference between sort of the influence of the genes there. So it is difficult now, particularly in the world of sort of pregnancy, with doing the research around something like MTHFR because of that huge sort of public health message with folate, and we know that it does protect against neural tube defects. So,
well, talking about ethics, what is the ethical line of what we as nutritional or natural practitioners can say with genomic testing, and what is a tall claim? Because, you know, I've heard in the news, you hear these people overseas getting lambasted for their claims, and you get a lot of orthodox medical practitioners burring up hugely about the use of genomic testing, and you know, they say it should only be, you know, used for this, for basically drug stuff. That, to me, is where the turf war term comes in. I just think they're not looking far enough. But what is the ethical line of what we can and can't say?
So for me, as a scientist who's worked in the area for so long and has not only read sort of so many papers, but has been involved in publishing papers and really been, I guess, you know, heavily involved in nutrigenomic research, I find that I'm very comfortable talking about the genetics. It is, when you talk about an ethical line, as someone who's a registered nutritionist, and I'm a geneticist, so I do work in clinical practice now in a private practice.
Yeah.
It's a hard line, because even when you're talking about things for the practitioners out there, even getting insurance, I write down that I do nutritional genomics, I do genomic testing, and they don't know what to do with that. It's a pretty new area.
Yeah.
But I think you can feel quite confident and safe when you are delivering information that is based on hard evidence and science. So it's not about saying, okay, because there are a whole host of, for example, if I can mention names, something like 23andMe, which is in the US, that just, you know, tests a whole heap of genes and a whole heap of SNPs. A lot of those are not based on hard science.
Right.
And that's when we start to get into some of these sort of confusing areas, because people are making assumptions, because we might know that, okay, this gene's involved in inflammation, or this gene's involved in detoxification. But even if the gene is involved in detoxification and you have found a genetic variation through, say, perhaps a company like 23andMe or someone else that just sort of does a lot of genetic testing that isn't necessarily based on sort of candid or chosen genes based on research, people can get confused and say, well, or they believe that because this gene's involved in a particular pathway and you've got genetic variations, that there's instantly some issue, an issue with the gene, or they might say, well, if you've got problems with detoxification, toxins are linked with cancer, and they might start to sort of make claims that aren't really valid or based on medical research. So that's where it gets confusing, and that's where, sadly, I think people, the whole validity and reputation of genetic testing can start to come apart. So the practitioner really needs to be cautious of the type of company that you use. You want to know that I obviously don't have a lab anymore. So I use genetic testing companies, and you need to make sure that it is based on, you know, good evidence. As particularly if you get Australian companies, they should be passed through TGA and all the regulatory boards and things like that. So if you are using a company that is based on research, and you wouldn't have to be an expert in genetics, you know, I don't want people to think they need to go out and do 15 years like I've done to be an expert, but just being able to look at a couple of papers or using a company that you know is based on research, and doing most of the companies too here in Australia, well, I should say all the ones that I think are approved by TGA and do things properly, require some kind of training before you're allowed to do the genetic testing. So by doing that genetic testing training and using a company that uses genes and tests SNPs or variations that are based on hard evidence, then you're not going to get into those particular issues where you're possibly making false claims.
Yeah. I leading on from there, I have to ask, and you can talk about this as being either the big brother question or the conspiracy theory question, because you'll get some people raising their hackles saying, well, hang on, if you've got a laboratory taking that test, by law they have to hold that test for a certain period of time. And the reason is because of legal issues. If there was an issue with the test that showed up later, maybe, for instance, we've heard of issues with pap smear tests in the past in Australia, that they were misread and not done properly. It's happened a multitude of times. So ethically, they have to hold on to that sample for a certain period of time. The conspiracy theorist, or they might have been watching too many X-Files or whatever, they might say, they're going to use that to give them, quote unquote, my gene information, and they'll be able to track me and all of that sort of thing. I don't propose that, but there are ethical considerations with regards to making money off genes. And this has happened with regards to the immortal ovarian cancer cells from a lady called Henrietta Lacks, who died, I think it was late 40s or something, 50s, from ovarian cancer. And today they use the HeLa, HeLa, ovarian cancer cells. They've recently won not recently now, it's probably 3 years old, they've won the court case to say that that's her intellectual property, if you like. So their family basically, So it's a storm of controversy. What are the legal requirements in Australia around storage of samples and use of somebody's genetic material?
Definitely, I'm not surprised that, when you talk about this case being won, because there's no way in this day and age that could be done, particularly here in Australia.
No.
So when we were doing our research studies and we wanted to test certain genes, all of this stuff has to go through ethics committees. So you submit the project, or even if you're a commercial company, you would still have to go through the right type of process, and you have to sort of have these things accepted. And they go through this with a fine-tooth comb. And then you would have to come up with a consent form when they've decided that what you want to do is appropriate or ethical or legal. Then there's consent forms, because with genetics, you need to have informed consent about what is going to be done with your DNA.
Yeah.
So if you are going to, for example, be involved in a research study, we would have a consent form that already would have been through ethics a number of times, depending on what they pick up from it. And then you would have this and you would give it to the patient, and you would describe what you're going to do with their blood and what you're going to test for. You can keep the sample. So this is from a research sense before I talk about a commercial sense. So from a research sense, you could keep the sample because perhaps it's going to fail, or, you might need to repeat it, but you could not use it for anything else other than that particular research study unless it was passed by ethics, unless you had a sort of a tick box or something where they could sign to say, I agree
you need
to
a
my
new consent.
sample being... Exactly. So you need to consent to anything that's going to be done, and that's a legal requirement here in Australia. From a commercial perspective, I have never had a commercial lab, but my understanding is, and I do use companies now, and I have to give my patients a consent form, so I see that's the same. So if I'm doing genetic testing, they have to sign, but again, there is actually something on the consent form that says, do you consent to your data being used for research? So yes, you're consenting to, you understand there's going to be genetic testing, but then certain companies that do genetic testing will ask you, do you mind if we keep it to use it for research? And then the patient or the client can agree or not agree.
Yeah.
And it's interesting when you sort of talk about these, examples of people wondering what the hell you can do with their genes. People do. So even from a research perspective, particularly males, I'm not sure why, more so maybe not with the blood DNA, but I was obviously working in fertility where we would also collect semen samples and things like that too, and we'd talk about, stuff like that. And the men particularly sometimes are like, what are you going to do with this? Are you going to grow? Are you going to grow a little me?
Yeah.
Are you going to do something? So, people are genuinely concerned.
Yeah.
So you need to understand what is going to be tested, and be able to, educate and inform that person in simple terms and get them to sign a consent. And nothing can be done with the sample unless there is consent. So in this case, I think, the example you gave where they were using the HeLa cells that have been used, in labs all around the world for science, my understanding would be probably because it was back in the 1940s, things were a little bit different. But now that would be very, very much illegal, and things like that should not be happening. I can't talk about in other countries where sometimes things can be a bit debatable, but here in Australia, these things are very much regulated and monitored. But it's important for the patient, anyone that is, or the client too, if they are worried about this, that they ask those kind of questions as well.
So I've got to ask then, what is the true value of nutrigenetic testing? What can these tests tell us that without them you won't get the information for, and you might be misled?
So I guess the power in the nutrigenetic testing, for me, there are some multiple points that I think I'll bring up to answer that. But the first thing that's coming to my mind is I'm just thinking about a patient I had yesterday. So a lot of it is answering the why. So for those that aren't coming to you just for optimal health, who are interested in their genes, they've actually got something wrong that perhaps mainstream medicine hasn't been able to fix, or they've sort of got chronic conditions and they want to understand why or what they can do. Sometimes the genetics really does help people understand, because this is quite an important thing. Yesterday I was working with a very intelligent lady who has struggled with her weight for a very long time. And, for example, she had a whole host of genetic variations with inflammatory genes that are predisposing her to be to chronic inflammation and having a sort of an exacerbated immune response. Plus, she had a whole host of genes in relation to fat metabolism and insulin, glucose, etc. So she is at risk of, being more inflammatory, which we know that links with weight gain as well. Plus, she sort of had all these other things in the fat metabolism gene. So she had come to see me after sort of 15 years of trying every single diet under the sun. She didn't have a weight problem initially, but what had happened is she'd actually had a very, very serious car accident and never really recovered fully. And I think that's probably what triggered her, possibly the inflammation, because now she also has pain in her joints and all these sort of kind of inflammatory things going on. She's got an autoimmune condition as well. But she had this accident, never fully recovered, sort of put on weight because she was sedentary for quite a while. She was in pain. And when she got a bit better, she tried to exercise, but continually had back problems, and sadly has sort of had this sort of up and down with her weight and tried every diet. So I spoke to her yesterday, and I couldn't give her the magic ingredient and appeal and say this is going to fix it or this is a supplement you need. But by educating her on the importance of not pushing herself too hard, because the inflammation was of concern. And I will also add, the genetics is not something I do stand alone. We also knew she had high CRP, which is a marker of inflammation and things like that, etc. But I was able to explain to her that genetically she is at risk of these things and that she is predisposed, and because of some of these lifestyle things interacting with her genes, this is why she's really struggling to lose the weight. And what she had been trying to do was go, with a personal trainer who'd been literally pushing her to the max and smashing her, and then she'd get sick and be out for a month or two, and then she'd go back and train really hard. And so to her, this is not the kind what you want to do. You need to be thinking low impact, small steps. Be aware. Some of the genetic variations I was looking at to do with leptin. I said, be aware that you do want to eat more than you probably need to. Just educating her about that. And if anything, I think what this does, it empowers people. So she just said she hadn't felt good about this whole situation for a long time because she thought people thought she was a liar because she really had at times been quite good with her diet. It went up and down, but she said people just think I'm a liar. They think I'm in the cupboard hiding, just stuffing my face. And I said to her, well, I believe that you're not. I said, it is important what you're eating, and you wouldn't have put on, she wasn't obese, but she's overweight. I said, you wouldn't be overweight if, you weren't putting certain things into your mouth, but you are, you do have to try harder than others. So I was able to educate her, empower her, give her some strategies to sort of help compensate for some of those genes. So I would say the power is in really sort of helping people understand. Again, when we talk about MTHFR, we want to talk to people particularly with fertility and folate. Things with detoxification. Some people are more sensitive. some people will put creams on their skin and react and don't know why. Yeah. So if we can talk to them and say, listen, you don't have the ability to break down certain chemicals and toxins,
so
that's going to make you more sensitive. Then they go, oh my goodness, so there's not something, it's not in my head. There's actually something genetic going on here, and I can work with this. I now know that I'm likely to be more sensitive to this, this and this. I'm going to have to avoid these things in the environment. So, for example, if someone's got genetic variations in the cytochrome P451B1 and 1A1, 1A2, things like that, we know these are involved in breaking down things like, car exhaust fumes. So if that person lives right near a freeway, we can say to them, this is no wonder you're getting these particular health issues, and we can see you've got high benzene. You're not breaking down the benzene because, one, you're exposed to high amounts being near a freeway, but you can't tolerate as well as perhaps your neighbour because of these genes.
Yeah.
So it's really about educating them, empowering them, and it's not a quick fix, and it's not a standalone test. It's something you do in or alongside other testing. So when I do a lot of sort of nutrient treatments, talking to people about diet and possibly supplements, but I would never base that just on the genes. We'll talk about, for example, vitamin D is a good one. They might have genes that mean they don't sort of process and activate vitamin D as well. Perhaps they've got changes in the vitamin D receptor, so they're not using vitamin D as well. In those cases, I'll talk to the person about the importance of making sure their vitamin D levels are up at a higher sort of range.
Yeah.
But before telling them to take vitamin D, I'd also get their vitamin D measured.
Yeah.
So you mentioned, what is the point of doing genetic testing? Can you find out this information without it? So, for example, you could possibly say to someone, well, you're low in vitamin D, you need vitamin D. But if you've got the power of genetics and say, your vitamin D actually might be borderline normal, but when we look at your genes and you see you've got genetic variations in the vitamin D receptor, and we know that you've got issues with inflammation, and we know that vitamin D can protect against chronic inflammation, we then might make that judgment by putting all of these things together along with the patient's symptoms and say, well, I'm very confident that you need more vitamin D. So it's really trying to get to that personalised medicine, what that particular individual needs.
It's such a it's obviously a huge area, and I think it is a complex area, and it really needs understanding. And I've got to say, Dr. Denise Furness, you are the perfect person to explain that complex understanding in easily understandable terms. Well, no, you do a really good job. And so I've got to say, would you mind if we continue this on other podcasts? Because there's so much to go into, and it's just, I could talk to you for like 3 hours right now. But
absolutely.
I don't think our listeners would appreciate it.
I think doing chunks will be good too, because I'm not sure if you get listener feedback, but maybe we can, get topics and things that people want to know more about and focus on those. And I'm definitely happy to do more and try to flesh a little bit of this out. And for practitioners who are wanting to introduce this into the clinic, I think these are the kind of podcasts that are good to help give them the confidence of why these tools and genetic testing is of benefit. How to communicate it with patients, and just learning more about an area that I think is obviously awesome.
Brilliant. So how about in our next podcast, I'll set this up for our listeners, we delve into the MTHFR SNPs, because it's something that's commonly known, but I also think that sometimes it's mistreated and potentially overtreated in certain individuals. So I'd love to go into the whys and wherefores and responsible treatment of those.
Absolutely. I think that would be very important to go through that, because I will say that brings me a lot of business from patients that have come from other practitioners that have been told certain things about MTHFR that may not be as accurate as it could be. So I think that's a really important topic to cover.
Denise, thank you so much for taking us through genetics, or I should say nutrigenetic testing 101 today.
Thank you so much. I will hear from you very soon.
You will. This is FX Medicine, and I'm Andrew Whitfield Cook. This podcast was proudly brought to you by the Bioceutical Seminar Series, Reprogramming Autoimmunity. This is Andrew from FX Medicine. We thank you so much for your support over the last 2 years. We'd really love to remain clinically relevant to your practice. So if you know of an expert in some area, please let us know. You can contact us on fxmedicine.com.au, Facebook or Twitter.